Flow cytometry is a technique that has been really useful in differentiating different types of cells, even separating them. There's So if you were to look at T cells underneath the microscope, you can't tell T cells, cytotoxic T cells from helper T cells. You can't differentiate type 1 and type 2 T cells apart just using a microscope. So if you wanted to count the number of cancerous cells in a leukemia patient or the number of T helper cells in a patient, then you can take a sample of cells, stain them with an antibody that's been labeled with either a fluorescent label or some sort of labeling technique, and then you run it through this nozzle, and it's a focusing thing. So it focuses them into a stream of single cells. As the light goes through, if it hits a fluorescent stain, it is picked up by a detector, and it can actually do a cell count on the number of cells of whatever it is you're staining for. So here's a way of putting this out and being able to read the information. It will give you a scatter plot so that you can actually use multiple different kinds of labels and tell the number of neutrophils versus monocytes versus lymphocytes, and that's just separating out the type. So there's a lot of these neutrophils here, a few of the monocytes, and here's some of the lymphocytes that are in there. So now, if we just concentrate on the lymphocytes, it can show you some differentiation between the types of lymphocytes that are detected.